Okay, well, I think now that we're a couple minutes past the hour, we'll go ahead and get started. It is my pleasure today to be introducing Dan Sherbenou for today's Robert Allen Memorial Lecture Symposium Series. Dan is Dr. Sherbenou is a physician scientist in the Department of Hematology or Division of Hematology here. He has a clinical and lab focus on plasma cell disorders. He has been here as part of the program when he joined and founded the Sherbenou lab in 2015. He's, in addition to being a really good clinician, he's a great scientist with a lab focus on translational work that utilizes patient-derived samples, bone marrow, to identify new therapies, to try to personalize understanding and approaches to current drug utilization, and that tries to sort of push beyond what we sort of know or are learning about available and future therapies for myeloma. So happy to turn it over to Dan to talk about drug-resistant subpopulations in patients with myeloma.
Thanks, Dr. Forsberg. Well, thanks for everyone for joining today. Happy to share some of the work we've been doing and researching of multiple myeloma. Today, I'll talk to you about our studies on drug resistance and personalized medicine in patients' samples from multiple myeloma. So our outline that I'll be going over today, so I wanted to give you an introduction into multiple myeloma for those of you who don't deal with this on a daily basis like Dr. Forsberg and I do. Introduce you to our platform. Second is this we call myeloma drug sensitivity testing, or MyDST. This is our approach to personalizing medicine for myeloma. Next, walk you through some drug resistance projects that we've been working on. First, we'll be talking about investigating immunomodulatory drug or IMiD resistance. And then I'll end with some immunotherapy discussion, first on CD38 monoclonal antibodies, and potential to retreat with that drug class. And lastly, talk about bispecific antibodies in MyDST and where we're going currently in the lab. So multiple myeloma is the second most common blood cancer. This diagram shows you the hematopoietic cell tree, starting with the hematopoietic stem cell and all the myeloid and lymphoid malignancies that can be derived from the different cells that are differentiated from the hematopoietic stem cell. So on the left, you have myeloid cells, which can lead to myeloid neoplasms, AML, and CML. On the right, the least mature lymphoid malignancy is acute lymphoblastic leukemia. Next, we have lymphomas, and then the most differentiated cell that leads to malignancy is, we call it multiple myeloma. So that's derived from the plasma cell. And right now, myeloma has turned into a pretty significant health concern in the US. The incidence has been increasing every year. It currently afflicts more than 150,000 people in the United States. So multiple myeloma, as I mentioned, is derived from plasma cells, which are unique cells in our body. They secrete antibody protein. That is their full-time job. So this is just an illustration of what a plasma cell looks like. It has very extensive endoplasmic reticulum, which is kind of the protein production factory that leads to the secretion of this Y-shaped antibody protein. Because it makes so much protein, it's very dependent on its ability to degrade the byproducts of that protein production through proteasome. And a lot of the unique treatments we use for multiple myeloma exploit these unique cellular attributes of these cells. So clinically, what's unique about this is we can follow the antibody that's produced by these cells because it's monoclonal, meaning that it's all the same. So when we run the patient's protein out of the gel, we call this serum protein electrophoresis, or SPEP. We can detect a monoclonal spike or an M spike in the gamma region most commonly, and that's shown here. This is an abnormal peak. The first peak here is albumin, and there are some other normal peaks in the other globular protein regions, but this normally should be a broad smear on the gel. When you see a discrete band, that's abnormal. And what that is just this Y-shaped protein that's made up of heavy and light chains in a patient that has myeloma, it's all the same, and that's why it's migrating as a distinct band. The other clinical test that we use is a tumor marker to monitor people's response to therapy and relapse, is the serum free light chain assay. So this is dependent on our ability to detect the small part of this protein, the light chain, which is in equilibrium with the heavy chain and can be detected at low amounts normally in patients, but in normal people. But when we have a patient with multiple myeloma, they make all kappa or all lambda from their myeloma cells. And again, this is a clonal process, so we can follow that in their blood as well. So those are our blood tests. The genetics of multiple myeloma are complex, to say the least. So there are many different genetic drivers and progression events that can happen in these cells. In general, these genetic events, which happen through a process of aging, not necessarily that the patient was born with, but mutations that accumulate over time. Most commonly, well, kind of in three different buckets here, we have translocations, gains and losses of chromosomes, and then point mutations. So the common translocations we see in multiple myeloma involve the heavy and light chain loci, which is basically giving the promoter that's highly active in these cells to an oncogene. So chromosomes break and they rejoin and produce high levels of proteins like cyclin D1, which drives the cell cycle, c-Myc, which is a strong proliferative signal, etc. Point mutations that happen in many other cancers also are pretty common in multiple myeloma. So P53 mutations or deletions in the case of 17p chromosomal loss, RAS point mutations, RAF point mutations, IDH point mutations. These are all, again, pretty common in myeloma as well. So these three different categories can kind of overlap, but really we kind of see three different buckets of events in people. So it's a really exciting area to be in right now as far as on the medical side. Treatments are really expanding greatly since 2000. We've really moved the needle quite a bit in improving outcomes for patients with multiple myeloma. Currently, the treatment landscape of multiple myeloma is undergoing a real transformation with immune therapies. So basically how this works is when you have a patient with multiple myeloma, it's generally incurable. That's the bad news. The good news is we have lots of different drugs that work well for it. So when a person first develops active myeloma, this first peak, we can put them very reliably in remission, and they stay in remission for multiple years. When they start relapsing, we go through other lines of therapy. And really in this lines 1 to 4 range, we're using combinations of drugs that include immunomodulatory drugs or IMiDs, PIs or proteasome inhibitors, and mAbs or monoclonal antibodies. And then more recently, CAR T or chimeric antigen receptor T cells and bispecific antibodies have been approved, and these are approved for people with 4 or more prior lines of therapies. And what's really going to be transforming over the next several years is that these are so good, they're going to be moved earlier, and we're going to start seeing combinations, different strategies as far as inducing and consolidating maintenance strategies to keep people in remission better and improve outcomes even more. So right now, currently, patients average greater than 10 years living with this disease if they go through an optimal treatment, including all these agents, autologous stem cell transplant, and Revlimid maintenance. But really, this is kind of a very rapidly changing area, and we expect to improve that even more in the next several years. So personalized medicine in multiple myeloma is a real strong interest of myself and my laboratory. This is kind of how we generally think of personalized medicine, most commonly in science and medicine right now. The Multiple Myeloma Research Foundation has this trial called the MyDrug trial, where they take high-risk patients, sequence their DNA to figure out what point mutations or what drivers have happened, and then put them on different buckets which include a drug that targets that underlying mutation. So this is a large multi-center effort, includes multiple different interesting new agents such as cobimetinib, which is a MEK inhibitor. And basically it starts induction with a treatment based on that along with dexamethasone. And then after 2 cycles puts them on a more typical IMiD, PI dexamethasone based regimen, continuing the targeted agent along with it. So data is starting to come out with the results of this trial, but it's been hard to accrue patients into such a complex trial. And so far the results have been a little mixed. So our approach is to take a different tact, and that is basically to think more about the phenotype than the genotype. So this, as I said, this study is a sequencing-based study. Our interest is really focusing on the phenotype of these cells, to going straight to their drug sensitivity versus resistant. What drug is working to kill the myeloma cells in that patient, and using that to base our treatment decision potentially down the road. So the way that we do that is myeloma drug sensitivity testing, or MyDST. And how this works is we take a bone marrow aspirate from a patient, which is basically a liquid biopsy. We put that in 48-hour culture, everything with an untreated control and every drug treatment done in triplicate, so that we can reliably compare at the end of the 48 hours. And our readout here is flow cytometry, and flow cytometry is basically where you run these cells through a machine, single file, and we can determine markers and isolate the myeloma specific killing effects of these drugs. And so the readout looks like this. So this is the myeloma population. It's kind of like bugs hitting a screen in this diagram. The viable myeloma cells, and then with your drug treatment, you can see those basically go away. And our goal here is within a week to give the physician a report that says your patient's susceptible to IMiDs, PIs, dexamethasone, chemotherapies, daratumumab, and this can help guide your treatment selection. That's the ultimate goal. So some background on what's better about this test compared to what everyone else has been doing right now. So we picked a 48-hour time point basically because although the myeloma cells can be hard to kill when they're in the patient, once we take them out of the patient, it's hard to keep them alive. There's a decrease in viability that occurs over time. We picked 48 hours because it's kind of when they transiently stabilize and we can reliably test drug effects. And that's shown in that first graph. Now we don't do CD138 selection, which is the most common approach to these type of assays in myeloma, because that really is detrimental to their viability without any drug treatment. So you can imagine it's hard to determine an effect if your untreated cells are not viable at the end of the culture, such as shown in the bar graph here. So we further improved this by humanizing the media to represent human plasma rather than normal laboratory cell culture conditions that you would maintain a cell line in. So that means physiologic levels of sugars, amino acids, all the things that all the components, nutrients that the cells are used to being in a native bone marrow environment. And so when we did this an initial study of 55 patients, we found that we could profile the drug sensitivity of patients from a newly diagnosed setting all the way to a multi-relapse setting. In this case, we studied people from diagnosis to up to 6 prior lines of therapy, which is more prior lines of therapy here going to the right. You kind of appreciate there's more green in the newly diagnosed setting, meaning more drug sensitivity and less resistance. Then we get to the 6 prior lines of therapy, people are pretty multi-drug resistance. And this recapitulates what we see in patients. So we're capturing that phenomenon in the lab, which is exciting. And this is another illustration of that. So people with we treat ex vivo with bortezomib or lenalidomide, which are basically our first line IMiD and PI treatments. We can see that generally there's this trend of drug resistance as you've had more prior lines of therapy with the patient that the sample was derived from. So when we looked in the subset of patients that we studied to see if our results could predict how people did, we learned a couple of interesting things. So Number one in the upper left-hand corner, in the newly diagnosed setting, if people get at least 2 drugs that they were sensitive to, here the middle column, they generally did pretty well. They either got to a CR and a few that almost got to a VGPR. Even better if you could give them 3 drugs, and in this group, all the people that got 3 drugs that they were sensitive to in our assay reached a complete response, basically. Now, in the relapse setting in the next graph, similar situation, although there's less drug sensitivity, as I mentioned in this scenario. So if you got 0 or 1 drugs that you were sensitive to, really, that you didn't respond at all or progressed, at least if you got 2 or more drugs that you were sensitive to in the assay, we could see that those people did pretty well. And we follow them over time, we see the same kind of effects. Getting more than 2 drugs in the newly diagnosed setting that you're sensitive to is predictive of doing better with the green line in the survival curve. And the relapse patients, same deal. Greater than 2 drugs, you tend to do better. And so when we combine the drugs that we're treating in the dish in single agent format, just mathematically, we can see that we can predict pretty reliably these people that are going to get at least a partial response from their drug treatment. So that we call these the true positives from the test. And we can also, on the other hand, identify the patients that are going to be truly negative, the people that aren't going to really receive a partial response or progress on their next line of therapy, and that's in the red quadrant up here. So overall, we had a couple, 1 or 2 false positives, false negatives. So our sensitivity and specificity for this test was 96% sensitivity, 86% specific, again, for partial response. So pretty decent first effort to show our test characteristics for this. All right, so kind of talked about myeloma drug sensitivity testing. Let's drop into some of the other things we can use for this assay besides necessarily just measuring drug sensitivity. So again, when we kind of go back to our diagram of how we do MyDST, there's a couple of tweaks that we can do. We can actually look for biomarkers of drug response in the myeloma cells, in addition to just how well they're killed. And so the ways that we do that are to shorten the culture time to 24 hours. This is a time period where there's not cell death happening for most drugs that we use, but we can see molecular changes. And so the readout becomes, your viable population of myeloma cells is still there, but we can look at molecular events that are happening in that group of cells. And so the first story I'll tell you is about looking at the Ikaros and cereblon proteins. So these are the drug response pathway for immunomodulatory drugs. So that's going to be our biomarker, our readout for this particular chapter of the talk. So this is a project that's been carried out by Lauren Davis, who's a PhD student in the cancer biology program here at the University of Colorado. So this is basically using MyDST to classify IMiD resistance. So I'm going to tell you a little bit about the nitty-gritty of this pathway and how it works. So basically, immunomodulatory drugs, this little orange oval here, binds to a protein called cereblon, or CRBN. CRBN is a component of a large protein complex called the E3 ubiquitin ligase. The job of that complex is to degrade proteins. When IMiD binds to it, it basically helps that complex recognize Ikaros 1 and Ikaros 3, which are myeloma survival factors, they're transcription factors, causes them to be ubiquitinated, and they're degraded by the proteasome when that happens. So the result of that is that Ikaros 1 and 3 go down. They're degraded with Revlimid or lenalidomide, pomalidomide treatment, and the cell undergoes cell cycle arrest and apoptosis as a result. There are downstream events that happen as well. So this event of Ikaros 1 and 3 going down, it also leads to the degradation of key proteins from myeloma cells called ER4 and MYC. And there are some off-target effects on in other immune populations that led to its original name of immunomodulatory drugs. So our hypothesis here is that if we develop a way to look at this pathway in MyDST, We can detect IMiD resistance, classify IMiD resistance by if we see Icaros 1 and 3 go down with IMiD treatment, and if we see ER4 and MYC go down with drug treatment. And so as background, what's known currently is that you can sequence the components of this pathway. The major thing that comes up is that you have cereblon loss of function, so that E3 ubiquitin ligase is lost. And there are several mechanisms that it can be lost. It can be spliced such that it's no longer functional. It can be totally lost by copy number lost. It can be point mutations or other structural variants. And through a couple of papers that have been done specifically Gooding and colleagues in Blood last year, the people with these cereblon abnormalities do worse than patients that don't. So if cereblon is mutated or otherwise affected, you wouldn't see the Icaros 1 and 3 go down because cereblon has lost its function to be able to do that. So, we developed this, Lorraine developed really this MyDST companion assay where she's looking at measuring IMiD-induced Icaros 1 and 3 degradation via flow cytometry. So how this works is similar to the parent assay. She takes unselected mononuclear cells, she adds this pomalidomide and then does flow cytometry, looking at just the myeloma cells, which are going to be these CD138, CD38 positive cells. And she can look at the Icaros levels she can look at the viability first to classify that these are sensitive or resistant. Now, in her assay, she's doing the same thing, but instead of 24 hours looking at these myeloma cells, and she can measure the Icaros 1 and 3 levels and see that pomalidomide is basically lowering those in this IMiD-sensitive scenario. And basically what we're looking for is, does it go down or does it not go down? In this graph, this is an example of where it goes down. So what she's found is that in patients, Icaros 1 and 3 are degraded by pomalidomide in the majority of patient samples. So she's got a cohort where we're looking at 20 plus patients, most of them in IMiD naive or just exposed category, but a few in this IMiD refractory category. And what she's seen so far is that the pathway is still active and functional in most of these people. So this was a little bit surprising because the cereblon data that I showed you before was published would lead to loss of this pathway going down. But keep in mind, there are 2 copies of cereblon and there might be some redundancy. You might have to have a full loss of function to really see that loss in the pathway. So we need more samples in this kind of IMiD refractory group, but so far it looks like we're only identifying 1 or 2 cases where you lose the ability to decrease Icaros 1 and 3 with an IMiD treatment. So that led us to think there might be some other pathway that can be exploited in IMiD resistance, so that you maybe in these patients, they don't care any longer that the Icaros 1 and 3 go down. They're able to survive on the activity of other survival factors. So Lorraine looked at the MMRF database, Multiple Myeloma Research Foundation Compass Project database, and looked at people that were pre and post IMiD. And what she observed was that MYC gene, targets of MYC gene expression tend to be upregulated in this IMiD refractory cohort. So her reasoning here is that maybe MYC is being activated independent of Icaros 1 and 3 going down, and so it's no longer dependent on that protein, but still dependent on MYC. And to evaluate this pharmacologically, she used a MYC inhibitor in IMiD refractory, focused on IMiD refractory patients. You can see a MYC inhibitor's dose response curve here, kills myeloma cells really nicely. It does have a little bit of effect on the normal hematopoietic cells, but much less, so there's a potential nice therapeutic window there. And when she looks at this group that's mostly POMID refractory patients, there's a nice waterfall plot of ex vivo response to a MYC inhibitor. So this is all suggesting that a MYC inhibitor strategy might be more effective in these types of patients. Conversely, what might not work is targeting the cereblon Ikaros pathway, again, with a new generation drug like iberdomide. So iberdomide is a much more potent version of pomalidomide and Revlimid, but it still works through the same mechanism by degrading Ikaros 1 and 3. So this implies to us that really the role for iberdomide going forward, maybe not necessarily in kind of the amide refractory patient, but ultimately maybe it should replace like frontline lenalidomide. So those are kind of the clues that we're currently operating on. Again, we need more samples, but that's kind of what we're currently, that's our working model. So I'm going to shift gears now to focus on the immunotherapy parts of the talk today. So talk a little bit about CD38 and daratumumab, monoclonal antibody retreatment, and then really an exciting area of bispecific antibody choice and relapsed multiple myeloma. So again, bringing it back to the assay, what else can we do with this type of platform? Specifically, we're going to focus on the immune cells here. And so our assay is unique. We don't CD138 select the myeloma cells away from their friends in the culture. All the T cells, B cells, macrophages, NK cells, they're all there in the cultures with the myeloma cells, kind of replicating the microenvironment or the other cells that are going to be present in the patient's bone marrow. And so we can utilize those they can be activated by different drugs like monoclonal antibodies or bispecific antibodies, and we can actually activate their killing of the myeloma cells and measure that in MyPST. So in our initial study, we did study daratumumab and found that it worked really well for this drug. So this waterfall plot shows you the ex vivo response to daratumumab in a group of naive patients or newly diagnosed patients, and a group of relapsed naive patients. Relapsed, newly diagnosed, both these groups are responding pretty nicely. As you can see, the bars look pretty good. This is again single agent daratumumab. In contrast, when you've been exposed and become refractory to daratumumab in the past, that group of patients really didn't respond, really at all in our ex vivo assay. And this is kind of this dotted line is just where we normally see significant results start to pop up, and really not much activity here. And we add this reagent called claudrinate liposomes, or CL, and which basically poison the NK cells, poison the macrophages in these cultures, and we can see basically the reversal of the sensitivity. So again, the monoclonal antibodies activating the immune cells in these cultures to kill the myeloma cells. And that's how it works in the body, and that's how it's working in the cultures. So we were pretty excited about this. So Olivia Perez Gacha, a former lab member, really kind of dived into answering a major clinical question we wanted to answer, which was, can you retreat a patient with daratumumab or isatuximab, the other CD38 antibody that's on the market, after you've been refractory? Maybe there's a treatment-free interval that you need to have in these patients. And that's exactly what she saw. So again, the daratumumab naive group responds really well ex vivo to daratumumab with at least about 60%, or I should say 40% decrease in their viable myeloma cells. In the group of samples that we looked from that have been obtained from patients off of daratumumab for less than 12 months, there's really not much activity of daratumumab at all in this group. For those who've been off of daratumumab for greater than a year, we start to see the sensitivity come back. So now again, our mean here of patients is coming back down below our sensitivity threshold at 80%. It's not as good as the naive setting, but it's a little bit encouraging. So to kind of look at a potential biomarker for why that's happening, we are seeing that CD38 level mirrors that sensitivity. So CD38 is the target for daratumumab on myeloma cells. Expression is very high in the daratumumab naive group. It's drastically reduced in the group of patients that have been off of daratumumab for less than 12 months. And it's coming back, not quite to pre-treatment levels, but it's coming back in those patients that are off of daratumumab for greater than 12 months, suggesting this may be why you can see a return of the drug effect. And I should note, this is basically a ratio. So we're looking at the levels of CD38 on myeloma compared to the non-CD38 expressing cells in the bone marrow. So 10-fold meaning, or 10 here meaning this 10-fold overexpressed, 100 meaning that's 100-fold overexpressed. So when we compare these two things directly in the same samples, see how they're related, we don't see necessarily a correlation, but what we see when we compare viability on the y-axis to CD38 on the x-axis is that there's basically a threshold of overexpression CD38 that you need to be sensitive to daratumumab, at least ex vivo. And this kind of makes sense, right? So basically there's no patients, maybe one outlier, but general no patients that have low CD38 that are sensitive. The ones that are expressing high CD38, it's a mixed bag. So it's necessary, but not sufficient for sensitivity. So you can see that about half the patients in this group are sensitive and about half are resistant. And this really goes well with what's published. There are other mechanisms of CD38 antibody resistance beyond just CD38 expression. So there are other, even though you have CD38 present, there may be other factors in some people that prevent the drug from working. So the next step really is to look at T-cell engaging of bispecific antibodies. The first bispecific antibody for multiple myeloma was approved 2 weeks ago. It's called teclistamab. And the way these drugs work is basically by acting as a bridge from T cells by binding to CD3 on the surface of T cells and myeloma cells via binding to numerous targets, including BCMA, FcRH5, GPRC5D, and CD38. And by proximity, basically attracting the T cells to the myeloma cells. That's how we see killing in these from these drugs. So this is a hot area. Basically every company is getting in on this, the potential of this treatment for multiple myeloma. As I mentioned, teclistamab is approved from Janssen. Elrantamab from Pfizer also looks very good. There are several other BCMA bispecifics that are looking positive in clinical trials. On the other hand, we have Symposia open here, targets FcRH5, has been really great for our patients in phase 1 even. Talquetamab as a GPRC5D bispecific, and then Sanofi has an anti-CD38 version of isatuximab, SAR 445527, which I'll call SAR in this talk. So on the flip side, although there's a lot of development on new bispecific antibodies, there's no biomarkers or companion diagnostics like MyDST at play. So we really wanted to kind of leverage what we specialize in for this topic. So to kind of mirror the CD38 monoclonal antibody retreatment question, there's also a big question right now, how do you sequence BCMA targeted CAR T cells with these bispecific antibodies? Should we be switching from one target to another? Can you get CAR T cells and subsequent line of therapies? At least anecdotally, it does look like from the few patients that have been published, like these two examples, you can give a CAR T a second time and still get into remission. You can give BCMA targeted therapies multiple times and get to remission. The second remission is generally shorter, but it can have potentially some clinical benefits for those patients. So we've been furthest along studying SAR, the CD38 T-cell engaging antibody. This is a unique molecule. It's actually trispecific. So in addition to targeting CD3 on T cells, it also targets CD28, which is a co-receptor to the T cell receptor on T cells. Targets CD38, much just like daratumumab and isatuximab do. There's a phase 1 clinical trial that's ongoing to study this in myeloma and lymphomas. This trispecific design, the rationale here is that we're trying to get, Sanofi, I should say, is trying to get a more potent T cell activation from this molecule. So that's kind of the context of why CD28 has been added. So Alana Keller is another PhD student in the lab, has been working with this molecule. Again, here's the structure. We can see a really nice dose response to this molecule, and these are in treatment naive patients. They haven't gotten daratumumab or anything. So this is basically looking to try and see what the expected effect in MyDST from ex vivo treatment with SAR is. You may recognize this is it's basically the most potent drug known to man in this, representing this class at very low picomolar concentrations down to 1 picomolar, 10 picomolar. You see a nice dose response from this drug. So it's in our assay, it's basically maxed out at 1 nanomolar. So we also have elrantamab that we have a grant from Pfizer to look at, similar for elrantamab. It's targeting BCMA, but we can see a nice dose response from this drug as well. Some patients respond better than others, and that's really what we want to tease apart. For these drugs, we know that they work. We're just trying to find out what patient best suited to treat them with. So Alana's hypothesis for this particular part of the talk is that post-CAR T myeloma is targetable with either a BCMA bispecific or the SAR bispecific, depending on maybe the CD38 versus BCMA expression, and that patient that's already been targeted with drugs that target both of those antigens. So she's accruing a population of patients that we're collecting bone marrow samples from before they go on CAR T and when they relapse after CAR T. And this is just the group she has so far. So in studying SAR so far, and we're picking 1 animal here as we expect that to give like a maximum effect. So if your patient is sensitive and not resistant to the SAR, it should show a response at 1 nanomolar. Again, this newly diagnosed patients generally respond pretty well to it, similar to daratumumab. However, in this post-daratumumab setting, we're seeing that there are more patients that are resistant to it and others that worked pretty well. And then in this post-CAR T setting, when someone's going to have had a long treatment-free interval from daratumumab, potentially because they've been on CAR T and maybe responded for a couple of years, we're seeing pretty good activity in the orange bars in that category. So this is encouraging that in the right patient, post-CAR T patient, we think in this case, SAR is going to work great. So some kind of interesting science behind playing with this molecule in MyDST. So in addition to sending us SAR, the Sanofi has also sent us null controls. One is lacking the CD38 binding capacity, or null CD38. One is lacking the CD28 binding capacity, so more like a traditional bispecific molecule that is null CD28. And so you see that in these superplots, basically, where each color represents a single patient samples, triplicates, SAR 1 nanomolar works great. For null CD38, there's maybe some variability here, but there's really no effect when you chop off the ability to bind CD38. CD28, loss of that, you can see it's still working, but it's not as potent as the full trispecific format. So this indicates that it's probably a good idea that Sanofi has been trying to push the area forward to see if we bind T cells more tightly, are we going to get even better effect? So another type of experiment that Alana's had some nice data on. She can compare this directly to the daratumumab to isatuximab, a monoclonal antibody format versus a bispecific format, which is better. So not surprisingly, the SAR is much more potent and seems to work better in these patients, and that's in the first on the left side of this graph. And this is a group that's pretty much daratumumab refractory. So daratumumab, isatuximab, not much activity in this group of patients. I should mention there's this, this is a 1 nanomolar for the SAR versus 20 nanomolar for the DARA. So even that 20x difference in the concentration, we're picking these concentrations again because we expect that to give us a maximum effect for the different drugs respectively. So indicating that this bispecific antibody looks like it is going to work better in a monoclonal antibody refractory population. So I showed you this kind of graph earlier for daratumumab. We're asking here, is the same story true for the SAR trispecific? So again, we're not seeing, there's a couple of potential exceptions, but in general, there's not many patient samples that are showing up as CD38 low, less than this like 6-fold threshold overexpression that we had observed that respond to the trispecific. So it's, and again, necessary to see trispecific work, and anyone that expressed more than CD38 in these red dots, very nicely sensitive to the trispecific. Interestingly, you're not seeing the same phenomenon of dots showing up in this upper right quadrant where they have high CD38. They're really not there. Everyone's responding if they have high CD38 so far. So that's kind of interesting. The mechanisms of resistance to daratumumab that I mentioned before that are CD38 independent aren't cross-resistant to trispecific or bispecific antibody format. On the other hand, when you have low CD38, yeah, you can have a low response to this type of strategy. So that kind of all makes sense. Further down that rabbit hole, basically, we can throw this SAR in and look at T cell degranulation through the proteins called CD107A and B, which are basically molecules that are expressed on the surface of T cells when they get angry. You can see that the SAR induces those molecules expression to go up. CD28 again does so a little bit, the CD28 null, excuse me, does that, but not as much as the others. And this is kind of how that looks on the flow plot. So basically we're looking for these T cells. These are the ones that are angry. These are the ones that are degranulating. Those are increasing with SAR. So again, similar to daratumumab working through macrophages in these cultures, it looks like the SAR bispecific, trispecific type of molecule is activating T cells as expected. And lastly, on this line of experiments, Alana did this kind of cool experiment with the CD38 null, where she's pre-incubating with the CD38 null and then throwing on the SAR full trispecific to see if that pre-incubation saturating the receptor, saturating the T cell receptor with CD38 null control is going to eliminate the ability for the SAR to come in and bind and activate T cells. And experimentally, she can see that that basically blocks the ability of this drug to work. So it needs to access the T cell receptor. The mechanism is holding true of its action. Once you block it, you really see that sensitivity go away. All just supporting that what we're measuring here is T cell directed killing, like you would see in the patient. So with that, I'm going to conclude by just telling you again, we are excited about MyDST. We think it's a useful platform for phenotypic personalized medicine, what you might call functionalized personalized medicine, and preclinical trials for patients with multiple myeloma. We have a clinical trial validation study I didn't have time to talk about, but it's through with partnering with Karyopharm. It's ongoing to evaluate MyDST's potential to be a treatment guide, basically to guide treatment decision making. And that clinical trial you can look up with this clinicaltrials.gov number. So Lorraine's project, I think, is showing that the MyDST and measurement of Icarus protein decreases following IMiD treatment. Ex vivo has the potential to identify a candidate, or what really we should be kind of doing with IMiD class, CELMoD class, versus targeting other pathways like MYC and IMiD refractory setting. So far, MYC inhibition looks like it's a promising strategy in IMiD refractory setting. Provocatively, maybe interestingly, there may be potential for CELMoDs to be the focus of that more be on early lines of therapy, replacing the less potent molecules in that drug class. Of course, that's our kind of ongoing opinion that we're going to continue to evaluate. So on the other hand, T-cell engaging therapies like bispecific antibodies can be measured with BioDST. We're really kind of trying to build this out and look at as many different targets and as many different molecules as we can. Ideally, it would be great to do a clinical trial here where we're doing the lab work simultaneously to treating patients on trials to determine if our test is able to predict response for patients going on those trials. We do think that CD38 can be recycled. So far, it looks like it's better recycled if you use a trispecific antibody format like from Sanofi's SAR molecule to exploit basically the recovery of CD38 that happens after a period off of it, such as in the CAR T setting. So with this, I'd like to stop and thank everybody that has contributed to this work. Of course, the people that actually have done all this work and made the data that I showed today. Zach Walker is senior research associate that's been in my lab since the beginning. And Lauren Ryman's our current technician that's about to go to MD PhD school. Alana and Lorraine I told you about with their projects currently in PhD studies. Sarah is our new technician and some past lab members that have contributed a lot to the data that I showed. Really should cite, of course, my collaborators, Peter Forsberg, Tomer Mark, the group at Cornell that send us samples, and my mentorship team here at the University of Colorado, which is consisted of Craig Jordan, Clay Smith, and James DeGregory. Of course, would also want to give a big shout out to our clinical team and our patients and the people that have funded this work. So I'll stop and thank you for your attention.
Well, Dan, that was a fantastic talk. Always great to hear about sort of the breadth of the different sort of exploration you guys are doing in the lab, over time. I guess I'll start it off, and then if anybody wants to ask a question of Dan, feel free to unmute and sort of jump in. I think it's set up for you to be able to do so, or you can put something in the chat, and I'm happy to ask along. Dan, you mentioned sort of evaluating samples with sort of the trispecific, including patients who are sort of post-CAR. I think one of the most sort of, eagerly anticipated question for us to sort of understand more around is how we sequence T-cell dependent therapies, whether that's, as we get more targets, as we get more therapeutic options, as things move from trials that have limitations in terms of patient eligibility criteria into the clinic, and we're sort of trying to expand on what we know and what we may not know. I guess, do you have specific thoughts about, or have you had any analysis that you've been able to do in patients, getting a bispecific, evaluated MyDST who've had a prior bispecific, or, it sounds like patients post-CAR are maybe, sort of showing good responses. I'm wondering if, other modalities that affect T-cell function, if you've had any chance to look at that at all, or if you just have any thoughts about what, the appropriate way to think about these patients in terms of, how we might sequence these therapies and what T-cell effects we might be, leading to.
Yeah, I think that's a great question, Peter. So, I think there's kind of two buckets for this patient population, kind of analogous to the IMiDs, where you're seeing either the pathway is still there or the pathway is down. In the case of bispecific antibodies or CAR T cells, there's a lot of research right now emerging that's showing that the same kind of thing is true. You can have target-dependent resistance, meaning loss of BCMA, mutations in BCMA, mutations in GPRC5D. That was kind of a big topic at ASH this past weekend, versus T cell exhaustion. So basically the T cells, especially from a bispecific antibody format, they get exhausted, they get tired, they're not ready to keep on responding to a T cell directed therapy. So I think we're really excited about using MyDST to kind of evaluate the underlying biology of these patient groups, pre-CAR T, post-CAR T, pre-bispecific, post-bispecific. As you know, some patients are getting CAR T first and then a bispecific. Some other patients are getting a bispecific first and then CAR T. So which is better? You know, what sequence is better is a major question. Is the T cell exhaustion just going to be too great from a bispecific antibody to expect that you would get good CAR T afterwards? And maybe some of that may in part explain why the results in that kind of post CAR T group have been, you know, have been a little bit underwhelming. So yeah, I think that anytime a patient at our center consents and is willing to donate samples, like going on a clinical trial like Symposia, we can look at their samples, and then when they relapse, we can look at their samples again. Not only focusing on the myeloma cells, but also focusing on what's happening with their T cells, and try, as we have been for the last 7.5 years, to learn as much as we can from the cells from our patients. So that's really the thrust of where we're focusing right now.
Okay, I guess I'll give a second for anybody else to jump in before I ask you more questions and pick your brain a little further.
Sure.
You know, I guess if anyone doesn't jump in, you know, another question I always, you know, like to hear your thoughts around is, you know, having done all this work on the CD38, you know, evaluations and DARAS, you know, sort of, or CD38 monoclonal sensitivity, I guess, what do you think is the most rational way for us to be using those drugs, you know, for us to be using CD38s, you know, in terms of duration, in terms of, you know, sort of things that might guide duration, and then, you know, in terms of patients where we may think about retreatment at you know, sort of future time points?
Yeah, I think that's another hot area, you know. So our study with this retreatment of CD38 really implied to us that after you become refractory to a CD38 monoclonal antibody, that your clinician should give you a break at least a year from that treatment. And that, you know, we had one particular patient that we studied where they didn't get continuous CD38 treatment, but actually went to stem cell transplant and then pomalidomide maintenance. They responded much, much better to retreatment than anyone else in our group of patients. So had a CR, still is a CR to this day. So that kind of, you know, got us thinking that really maybe we're treating patients with daratumumab or isatuximab wrong by continuing it all the way to progression. Now, that's just one patient, and the flip side of that is, like, if you look at the Griffith study data, when they give newly diagnosed patients a quad with daratumumab and Revlimid and Velcade dexamethasone, they get a really good response. And if they continue daratumumab with Revlimid versus Revlimid alone in the maintenance setting, patients continue to, well, in both arms, granted, patients continue to get MRD negative. The percent is slightly better in those people that get longer portions of daratumumab. And so I don't think that's probably statistically significant, but it is suggestive that daratumumab is right for patients in a longer time frame, especially in an upfront setting. So I think that's kind of an ongoing area that we'll find out more about. But really kind of that's one area where I really kind of apply MRD testing. So if you treat a patient to MRD negativity, you know they're going to do great. If they're high risk, you get them to MRD negative state, they're pretty much going to go to like a standard risk scenario. So that MRD negative is the goal. If people are MRD positive, you want to kind of keep their foot on the gas more. So I think in my practice, and this is kind of an evolving area, and certainly just my opinion right now, but I think that those people that get MRD negative would be a perfect candidate to just put on Revlimid if you had induced them with a quad. Whereas someone that's certainly high risk and MRD positive, that would be a patient that I would keep the foot on the gas and continue daratumumab longer. So thank you for the question, Peter.
Yeah. Sounds good. I think, always interesting to sort of hear thoughts about how we might also use risk and MRD to sort of adapt our approach. I know that, lots of different studies going on, hopefully in that sphere, even if it's sort of, baby steps to try to get kind of coherent data. But I think a lot of times now we're sort of just left, how do we apply that data with our best judgment to try to come up with the right approach for given patients and given circumstances. So I guess if no one else has anything specific, I'll say thanks, Dan. What a fantastic talk. Really exciting data, and really always exciting to hear about the way you're sort of trying to come at these questions that are really, germane to how we approach patients on a day-to-day patient basis. So thank you.
Thanks, Dr. Forsberg. Have a good day. Thanks, everyone.